Hello, biology. Purpose of this video is to give students who need to catch up on some of the 10-2 class notes an opportunity to do so, or maybe a student was absent and give them an opportunity to obviously get caught up. A couple reminders. Please start your numbering at number 6. If you are doing a Google Doc, let me get to my packet. If you are doing a Google Doc, we are going from number 6 to number 19. So that'd be 6 to 15, 16, 17, 18, 19. You'll need 14 rows, and you will need, obviously, 2 columns. Okay. For those of you that are doing it by hand, just draw a line down the center of the page. Remember, all class notes need to be in Cornell note format. Questions on the left and answers on the right. Because this is a video and you can pause, fast forward, rewind, I will not be spending as much time on each slide as I do in class. So what I will do is tell you with a finger pointing that I'm getting ready to bump up to the next slide. So if you're listening to music or you've got your volume down, you will still know when I'm planning on going on to the next slide. All right, let's get started. Okay, so eukaryotes, remember eukaryotes or eukaryotic organisms, those are organisms that have a nucleus. Their DNA is protected within the nucleus. A prokaryote, these are primitive eukaryotes are complex organisms with complex cells. Prokaryotes are single-celled organisms and their DNA floats freely in the cytoplasm. So they do not have a nucleus. These are primitive, less complex organisms. But there are 2 stages of cell division. Basically, the division of the nucleus, or the division of the chromosomes, or the division of the DNA, and we call that mitosis. And then the second type is a division of the cytoplasm, which comes right after mitosis, is when the cell, if it's an animal cell, it pinches inwards, and now we have 2 complete daughter cells. Plant cells, it's a little bit different. Plant cells are more rigid, and they can't pinch, but we'll get to that at the end of these notes. So cytokinesis is division of the cytoplasm, and mitosis is division of the nucleus or division of the DNA. All right, moving ahead. All right, we're going to abbreviate chromosomes as CHRs. So the reason why you are a combination of characteristics or a combination of traits of your mother and your father is because half of the DNA in all of your cells that has DNA, and some cells in the body don't, came from your mom, and the other half of the DNA came from your dad. And depending on whether or not that DNA codes for proteins or traits that are dominant or recessive, that will determine why you look more like your mom, why you look more like your dad, why you look half and half. And we'll get into that when we start talking about genetics. So before cell division, the chromosome gets duplicated or replicated or copied. That happens during the S phase of the interphase. Interphase is G1, S, and G2, and that's basically preparation for cell division. And it really makes sense for us to double the DNA or double the chromosome number because if you want each cell to have exactly, each daughter cell to have exactly what the parent cell had, you have to double it. If you don't double it, you end up with half of what the parent cell had. And remember, DNA are instructions for what a cell needs to do or might ever need to do. And you have to give it all of those instructions to make sure that it is capable of doing that. Moving on. All right, chromosome parts. So we call this half of the X a chromatid, this half of the X a chromatid. It's connected by this dense region in the middle called a centromere. And the reason why we call them sister chromatids is because they are identical to one another. So just to clarify, when we see these X's, okay, we refer to these as chromatids. But in anaphase, when these two get pulled apart, we then refer to them as chromosomes again. Okay, so after they've replicated, they're chromatids, and we call them chromatids all the way up to anaphase when they get pulled apart, and then we call them chromosomes once again. Each pair of chromatids is attached to an area called the centromere. During cell division, the chromatids get separated, and each new cell, which is called a daughter cell, gets one chromosome. Now remember, humans, we have 23 pairs of chromosomes, okay? 22 of those are autosomes, and then the other two are sex chromosomes. The X and the Y chromosome, if you have an X and an X, you are a girl, and if you have an X and a Y, you're a boy. I think I just saw Hud. You want to go see Mom? Come on, bud. Come on, Hudson.
Go up a little bit, sure.
Come on.
Tell me when.
Okay. Sorry. Come on, out. Gave you a chance. Oh, you're very lucky. I never have Hudson sightings in my videos. Of course, he wouldn't want to hang out with Dad. No, he loves his mom way more than his dad. Okay. So chromatids separate, and each new daughter cell gets one chromosome. So obviously, if this is happening in humans, and all the diagrams that you see are not humans, that would be way too complicated because we'd have 23 of these pairs separating, and that would be really difficult to draw. So we're just keeping it simple. We're just talking about one chromosome pair. Moving on. A cell cycle is a series of events that cells go through as they grow and divide. You're going to see that definition on the test. Make sure you know it. There are 3 parts to the cell cycle. Interphase is basically preparation for cell division. Interphase is when the DNA doubles or the DNA replicates. And remember back to our last little mini unit, DNA replication is when you would have a mutation. And if that mutation causes a protein to be non-functional, especially an important protein to be non-functional, that could harm the organism. Okay, but mutations lead to new characteristics. Maybe that characteristic gives the organism an advantage in its environment, which leads to evolution. So mutations are not always bad. Main events of the cell cycle: the cell grows. As you remember back to section 10-1, you want kind of this happy place in between the right amount of volume with surface area. You don't want too much volume, and you want to maximize your surface area. So if the cell gets too big for that, it knows it's time to divide. And then it does a preparation for division. That's when we're going to be doubling the DNA, doubling the chromosomes. And then during mitosis and cytokinesis is when we actually form 2 daughter cells. Wearing my Mets gear today. This he doesn't look like this anymore, but this is Jake DeGrom, who I've read many articles saying he is the best pitcher in Major League Baseball. Unfortunately, we don't have a couple of him, so we can actually get to another World Series anytime soon. Sorry, I digressed. Moving on. Okay, phases of the cell cycle. So you got the G1, you got the S. That's when DNA replicates. You're going to need to remember that for the test. You got the G2. And then the M phase is actually mitosis. Get some of my mess cleaned up here. Moving ahead. Okay, so this is, I really like this diagram because I'm a visual person. I mean, you can tell me words all day long, and until I spend, hours memorizing them, it doesn't click. This kind of makes a little bit more sense to me. So remember, interphase, okay? Interphase is the G1, S, and G2 represented right here. DNA replication happens during the S phase. This is when the cell is growing, okay? This is when it's getting ready for mitosis. So the centrioles are forming, the chromatin starting to condense into visible chromosomes. And then you enter mitosis. And of course, we got prophase, metaphase, anaphase, telophase. Remember, cytokinesis is part of cell division. And you'll notice cytokinesis. The only thing I don't like about this diagram is that it has cytokinesis starting during anaphase. Actually, it starts during telophase. So just pretend the line for cytokinesis is right here. I don't see any diagrams, pictures with the cell starting to pinch during anaphase. It's always during telophase. Okay, so cell division, mitosis, PMAT, PMAT, or TAMP, if you want to remember it backwards. And then cytokinesis is when the cytoplasm splits, and then you got $2 cells, and then the whole thing starts over again. Okay, please use this as an opportunity to practice with your notes. Some of you are going to fast forward. The more you practice with your notes now, the better prepared you are for the test when it gets here. So 2 stages of cell division. Take a look back at number 6 if you're not sure. You got it. Mitosis and then cytokinesis. Oh my goodness, Paulus, are you kidding me? Let's see. See if you can think of what number 1 is. Yep, E. It's genetic information. It's basically organized DNA. Each one gets duplicated. That was the big clue there. Chromatid. Yeah. F. 2 chromatids, 2 sister chromatids make a chromosome. Good. All right. Centromere. Where's that? I almost gave it away. A. Middle of a chromosome. Good. Cell division. C. Good. Mitosis and cytokinesis make up cell division. Interphase. D. Period of growth between cell divisions. Oh, beg your pardon. Yeah. And then the cell cycle should be, cell cycles G1, S, G2, and M phases. All right. Good. Moving on. Okay. Phases, like I said, PMAT, prophase, metaphase, anaphase, and telophase. You guys are welcome to abbreviate them just with the first letter. Okay. So PMAT, or if you want to memorize them backwards, TAMP, TAMP. To tamp something means to flatten it. Moving ahead. Okay, prophase, what's going on here? So the centrioles, so these things in the book, they kind of look like little red pieces of licorice or little licorice bites. These are the centrioles, okay? The spindle kind of looks like a spider web. Spindle fibers. You'll also hear it referred to as microtubules. All right, so the centrioles separate, okay? So the centrioles get replicated also during interphase. So now there's 2 pairs. One pair goes to one end of the cell, and the other pair goes to the other end of the cell. Okay, so they take up positions on opposite sides of the nucleus. Chromatin condenses into chromosomes. So remember, chromatin is just DNA wrapped around those histone proteins, which is almost the least organized way you can have DNA. And then you get nucleosomes, and then you get coils, and then you get supercoils, and then you actually get the chromosome. So the chromatin condenses and organizes into visible chromosomes. Remember, DNA has been replicated. So in this diagram, it's representing 2 pairs of chromosomes. So chromosome 1 and chromosome 2. Of course, we have 23 pairs in our cells. Centrioles separate and a spindle begins to form. So this is the spindle. Eventually, it will go all the way across the cell. Nuclear envelope, which is a fancy way of saying the nucleus membrane or the nuclear membrane. An envelope has things in it. That's why they call it the envelope. All right, so the nuclear envelope begins to break down. That's why you see it dotted here. And remember, when we have the X's, we refer to them as 2 sister chromatids, but each chromosome is made of 2 sister chromatids. So here, all the way up to anaphase, we refer to these as sister chromatids. Moving ahead. All right, metaphase. Meta is Latin for middle. Okay, so this is when the chromatids line up in the middle. And I understand I'm still referring to them as CHRs. Okay, centromere, by the way, should be pointing to this black dense region in the middle. Spindle fibers correctly identified. Centrosome is the same as the centriole. Okay, so the chromosomes line up in the middle. Microtubules, which is the spindle. Microtubules connect to the centromeres. That's how these sister chromatids are going to get pulled apart eventually. Of course, in the crash course video on mitosis, you learned inner details about how the chromosomes get lined up in the middle. Protein dynein is involved. Motor proteins are involved. You don't need to know that level of detail for my exams, but if you ever take AP Bio, you'll need to know that. You get into pretty much AP Bio gets into the nitty-gritty molecular level of all the high-level stuff that you learn in biology. But of course, I hope you take my AP Environmental Science class before you take AP Bio. It's easier. It's a great AP Science class to take when you're a junior because you're just getting your feet wet, learning about AP process. It's a fast-paced class. I'll agree. It's fast-paced, but kids seem to like the class. I have about 100 kids taking that class in 3 sections every year. Sorry, little plug for my AP Environmental Science class. You're going to be meeting with your counselors virtually over the next couple months. Think about it. If you did good in bio, did good chem, take AP Environmental Science. Now, actually, if you're a freshman, you'll be thinking about that next year. Sorry, my bad. All right, moving on. Okay, please practice with your notes. What are the 4 phases of mitosis in order? Take a look back at number 13, but you don't need to take a look back at number 13. You know the answer. PMAT, prophase, metaphase, anaphase, telophase, or backwards, TAMP, telophase, anaphase, metaphase, prophase. Okay, what's one thing that's going on during prophase? Take a look at number 14 if you can't remember. So the centrioles are starting to move towards opposite ends of the cell. The nuclear membrane or nuclear envelope starting to break down. Spindle fibers are starting to form. Chromatin has condensed into chromosomes, visible chromosomes, which are now called 2 sister chromatids. Good. What's going on during metaphase? Take a look at 15 if you can't remember. So that's when the 2 sister chromatids, were a chromosome, line up in the middle. The spindle, which is also referred to as microtubules, the spindle fibers, attaches to the centromere, that central dense region of the chromosome. The centrioles are on opposite ends of the cell. Chromosomes, chromatids line up right down the middle. They're getting ready to get pulled apart. Moving ahead. Okay, anaphase. All right, so anaphase is when the sister chromatids get pulled apart from one another. Okay, once they are pulled apart from one another, they are referred to again as individual chromosomes. As you can see, so the centriole, okay, on opposite ends of the cell, We got the mitotic spindle, which is the spindle fiber. It's a fancy word for the spindle fiber. We got the cell membrane. And then, of course, these are chromatids, which are now referred to as chromosomes, are getting pulled apart. The reason why you kind of see them in this V structure, the centromeres are attached to the microtubules, the spindle fibers, and they're being pulled by the centriole. But remember, this is cytoplasm, which is kind of a jelly-like substance. So there's friction. The ends of these chromosomes are being dragged kind of through this jelly. And that's why they're kind of lagging a little bit behind. Most of the drawings you guys see in the book or online have it represented like this. Chromosomes move until they have separated into two specific groups. And basically the bottom line is they want to get these chromosomes as far away from the center of the cell as possible, because eventually the cell is going to pinch in here in the middle. You don't want chromosomes anywhere near the pinch, because if a chromosome ends up over here, so this one ends up with 3, this one ends up with 5, that could be a real problem for the organism. Definitely a real problem for the cell. Okay. All right. Moving ahead. Telophase. That's when the chromosomes gather at opposite ends of the cell and they lose their shape, so they start to convert back into chromatin. And again, we're now calling these not sister chromatids. They are no longer related. They are individual chromosomes in each new cell. You start to see the cell pinch. Animal cells do this. Plant cells do something a little bit different. My son's alarm's going off. Bummer. This is called a cleavage furrow. Okay, cleavage furrow. I'll have you guys draw that when you do your mitosis drawings. A new nuclear envelope is forming, which is important because now these chromosomes need to be protected. The DNA needs to be protected inside of the nucleus. Well, let me holler at my son real quick. He heard it. All right. Also, a few things just to know, but you don't necessarily have to write them down. The spindle fibers are starting to break down. Obviously, there's no more need for them because they already pulled the chromosomes apart from one another. And then the centrioles are also dissolving because they are no longer necessary. Centrioles are kind of like a fishing pole, and the microtubule spindle fibers are kind of like the fishing line. The centrioles actually are pulling the spindle fibers towards them, and spindle fibers are attached to the chromatids, chromosomes, and they're pulling them towards each end of the cell. And of course, on the other side, it's going to be going that direction. All right, moving ahead. Okay, so cytokinesis. So this is the second part of cell division. This is cytoplasm, division of the cytoplasm. So it pinches in half. And again, this is called the cleavage furrow. Again, these are individual chromosomes. Cytokinesis technically starts during telophase. Most drawings of telophase you see have the pinch beginning. These are referred to as daughter cells. Again, we have the nucleus. So the nucleus or the nuclear membrane is completely reformed. Each daughter cell is an identical set of duplicate chromosomes. So in this case, we're talking about 7. So it's got 7 individual chromosomes, which is about a third of what humans have. All right, moving ahead. Okay, so cytokinesis is different in plants. There is a carbohydrate. It's called cellulose. Cellulose is actually in the cell walls of plant cells, and it makes them more rigid. And evolutionarily speaking, the reason why that's important is because if plants aren't getting enough water, as you know, they end up drooping and then they die. But it helps the plant stay upright longer because remember, if it's upright and if it's outdoors competing against other plants to get sunlight, it really needs to stay upright as long as possible. So the rigidity or cells being rigid enables them to stay upright for longer when they're not getting enough water. But also prevents the cell from pinching inwards. So what happens in plant cells is it produces this structure called a cell plate down the middle, and then eventually a cell wall will form on both sides of this. And we will then have two daughter cells. So it doesn't pinch, it just kind of forms these structures down the middle. So that makes plant cells different than animal cells. Moving ahead. Okay, what's going on during anaphase? Take a look back at number 16. Good, so the sister chromatids, which are now referred to as chromosomes, are getting pulled apart from one another. Good. Let's see, also during anaphase, chromosomes are moving into two groups as far away from the center as possible, because remember, in the center is when the cell eventually is going to pinch inwards, and you want to make sure that each daughter cell ends up with the right amount of chromosomes, the right number of chromosomes, identical sets of DNA. That's the goal. Telophase, take a look at number 17. Yeah, so the chromosomes are gathered at opposite ends of the cell. The centrioles start to break down, spindle fibers start to break down, but the nuclear envelope or the nucleus membrane starts to reform. Remember, membranes protect the DNA inside of the nucleus. That's one of the characteristics that makes eukaryotic cells different than prokaryotic cells. Also, chromosomes start converting back into chromatin, less organized. The only reason you need chromosomes in the first place is to make sure that each daughter cell ends up with a DNA. Could you imagine having this huge ball of chromatin the DNA is double, it's all tangled. Try separating that apart and making sure that each daughter cell gets the exact same amount of DNA. So they form into chromosomes to make the process of separating it easier. Okay. All right, what two things form in plant cells that do not form in animal cells during cell division? What are those two structures that plant cells have that animal cells don't? I'll give you a clue, they form down the middle, creating a daughter cell here and a daughter cell here. So the cell plate first forms, and then the cell wall forms around the cell plate. And again, plant cells don't pinch because they have cellulose, which makes their cell walls rigid, so they can't actually pinch inwards. Animal cells do not have cellulose, so it's a lot easier for the cell membrane to pinch inwards. Moving on. Okay, some practice questions. Series of events that all cells go through as they grow and divide. What do we call that? Yup. Cell cycle. Phase of mitosis, during which the chromosomes line up across the center or middle. Whenever you see center of the cell, you should replace it with middle, and it makes it a lot easier to remember. Yeah, middle, metaphase. Good. All right, cytokinesis usually occurs. When does cytokinesis occur? Yeah, it actually starts during telophase. Good. During mitosis, sister chromatids separate from one another. What phase are the chromatids pulled apart? And then they are referred to as chromosomes. Yep. Anaphase. Nice job. All right, ladies and gentlemen, that is it for the 10-2 class notes. We will see you in the Zoom.